Okay, hello everyone. This is Michele Rossini from the Finnish Museum of Natural History. It is a great pleasure to be here with you, and today I'm going to introduce you our new research project on Malagasy dung beetles, and I'm going to talk about the findings of our recent expedition in Mauritius. So our research aims to investigate the mechanisms that drove the evolution and radiation of Scarabaeidae dung beetles in Madagascar and Mascarene Islands. And basically, with this study, we are going to explore how different types of speciations, bioclimatic, biogeographic, and anthropogenic factors contributed together to the diversification and extinction of dung beetles in this region. And extinction fits very well mascarene biotas, as many species have gone extinct over the years, including dung beetles. Indeed, subfossils remains have been recently found in Rodrigues and Mauritius. Anyway, in order to answer our central question, we are going to combine dated phylogenetic reconstructions and novel statistical modeling of species range evolution. And of course, we are going to use dung beetles as model organisms, given that they exhibit an impressive high rate of endemism in these islands. And recently they suffered extinction in the Mascarene Archipelago. So two main expeditions were programmed for the next 2 years. The Mascarene Archipelago for the current year to recollect dung beetle species only known from a few specimens collected a long time ago. And of course, to look for additional subfossils remains of Scarabaeidae to include in our study, while for 2022, Madagascar is going to be the objective of our field trip. And, but things unfortunately changed because of the COVID-19, and we were forced to focus our expedition just on Mauritius, the only island of the Mascarene Archipelago that to our knowledge still has 5 endemic dung beetle species, I mean still alive species, 4 Nesosisyphus plus the monotypic genus Nesovinsonia. Anyway, before getting to the field, we had to pass through the 14-day quarantine, which was terribly mind-numbing, but at least we got some reliefs from local beers and our dear fellow, the flying dodo, which gave us great shots of the beauty of the island. But eventually we were released and we could finally start our field trip. So briefly, some details about the Mascarene Archipelago. So Reunion, Mauritius, and Rodrigues are the 3 volcanic islands belonging to the Mascarene Archipelago. They are located in the middle of the Indian Ocean, east of Madagascar. And with each 8 to 10 million years old, Mauritius and Rodrigues are the oldest islands, followed by Reunion. And like many other oceanic islands, their ecosystems are very fragile, and the arrival of Westerners at the end of the 16th century had a devastating effect on mascarene biotas, as many species went extinct. And over the years, Mauritius lost about 98% of the native forest, which are today replaced by a huge number of invasive plants from South America, Asia, Africa, and so on. But also numerous are the invasive animals that had a terrible impact on native ecosystems. From a climatic point of view, Mauritius is definitely wetter in the east than in the west, but in the western part, Mauritius is characterized by a higher number of microclimatic conditions, and as a consequence, multiple microhabitats and a high rate of endemic animals, including our beloved dung beetles, which are basically distributed along the western part of the island. So we set up pitfall traps in several of the places where the 5 endemic species of dung beetles were historically collected, the blue labels in the map. that is the Malta Mountain Range, the Black River Gorges National Park, and where we set up traps in 2 conservation management areas in Marlong and Montfaucon, but also in private areas in the eastern mountain range like Montagne Créole and Vallée de l'Est. So since 3 of the 5 endemic species are wingless, our sampling included also the sifting of the leaf litter of some of the visited forests, such as the upper forest of Lepus Mountain and the forest preserved within the CMA in Kokok Mountain. And as for the subfossil survey, we used a Russian corer, and each soil sample was split into 10 centimeter deep slices. However, these samples have yet to be processed, but anyway, coring was carried out in 3 localities, the ones indicated in the map with the dinosaur. So Marlachaux, which is a swamp in the middle of a sugarcane field. Ile d'Ombre, which is an island located in the northeastern part of Mauritius, and Kanaka crater in the southern and central part of the island. So, well, collecting those 5 endemic species was absolutely more challenging than expected. So our traps consisted of a jar buried in the ground, partly filled with 94% alcohol, and the bait was hung over the jar. Noteworthy, in the past, most of these species, most of the endemic dung beetles from Mauritius were collected with chicken dung, and the only Nesosisyphus pygmeus showed high attractiveness to monkey excrements. Well, at the beginning, we tested the attractiveness of these beetles to different baits, as you can see from the picture. But only chicken and human dung brought some endemic dung beetles. So we decided to reset up our traps, eliminating alcohol and placing the bait, only chicken and occasionally human dung, directly in the buried jar. And this new setting was definitely much more successful in terms of number of specimens and species collected. So as a result, 4 of the 5 endemic Mauritian dung beetle species were recollected, such as 3 out of 4 Nesosisyphus species. The only Rotundatus misses in our list. And importantly, 4 specimens of the legendary Nesovinsonia vinsoni, which was finally recollected after 80 years. And that was really, really exciting for us. And now finally, we have good specimens for DNA analysis. So again, only traps placed in high elevation forests at about 700 meters above the sea level allowed us to recollect our endemic dung beetles. But consider that the highest peak in Mauritius is about 830 meters. And also very interestingly, peatful traps located 10 to 20 meters away from the punctual collecting localities brought 0 specimens. Does it mean that these beetles are so sensitive and demanding in terms of habitat? This population really so localized? Who knows? So many questions are still unanswered about these amazing beetles. And also quite interesting, some specimens of Nesosisyphus regnardi were collected in peatful traps baited with carrion of land snails. But anyway, the most puzzling thing is that the 4 Nesovinsonia specimens came exactly in the same peatful trap, which was placed about 10 meters away from the other traps with the same bait, within the same forest characterized by the same vegetation, and so on. So is really Nesovinsonia coprophagous beetle? Probably not. Perhaps it came occasionally to dung, but its biology, yes, is still very enigmatic to us. And as regards sifting, it brought us just some specimens of Nesosisyphus regnardi and Vicinus, but sadly no Nesovinsonia specimens were captured with these collecting methods. That's all, and thank you so very much for your attention. And I would like to thank especially these institutions that significantly supported both economically and logistically our research and field trip in Mauritius. And of course, if you have any question, I'll be very happy to answer you. So bye bye.